that it's microbial. Um, we do know that the undifferentiated is microbial, so it's not plants, it's not worms. Um, but yes, there are other groups here in the microbial world. It's not just protozoans. I mean, you've got nematodes, so some of this could be nematodes. There, there is a huge amount of research out there on PLFA. The the original extraction method was developed in 1953, and it hasn't changed much. The issue is that there's so much argument in the scientific world about which fatty acid is a biomarker for which organism. Part of the struggle is understanding the environment in which the sample was taken. So here's a great example. The biomarkers used for actinomyces are the same biomarkers you use for desulfobacter in lake sediment. So desulfobacter dominates in saturated environments, especially anaerobic saturated environments like ocean beds and lake beds, but they do not dominate in your soil and in agronomic soil. Actinomyces does. Now that doesn't mean that there's not any desulfobacter anywhere in your field, right? But we do not list that biomarker as desulfobacter because 99% of the time it's not. It's actinomyces. So there is argument about it. We have modified our biomarker list a little bit based on new research, and we tend to, when I did my literature review, it was like, okay, if 80% or more of the people who are publishing on this agree, we're going to use that one. If they don't, we just left it out. If that helps, makes sense. Yeah, it's not a perfect science. James asks, is there a key that was sent to us that corresponds with the field ID so we know which test refers to which sample location? I'm not sure on that, James. I don't have that information. Everything I see at the lab is literally just coded and it's by design. It's so I don't have any bias on what I think should be or shouldn't be. So Jonathan and the team at Ecodiasis have done a really good job of keeping me in the dark. Um, Do I have any data and results about nematodes and PLFA? I have not seen nematodes tested with PLFA, and the reason is because the biomarkers don't exist. Um, at least not good ones. There was argument about it quite a while. The problem, nematodes are actually a higher level organism. They're the smallest animals on Earth. So they end up with a huge wide range library of PLFA, and it's hard to differentiate those PLFA all the time from plant root because now we're getting in that category where they're all kind of complex. Um, there are many other techniques that are better for nematode testing than, and they're getting better all the time too. Everyone focused on cis nematode, that was it. And now there's some groups out there that are doing free living and root association nematodes, things like that. I've been approached by a few of those companies. We haven't adopted it yet simply because I don't have time to implement a new test right now, but we are looking at it. It's a good question. Um, all right, I'm going to share a screen here, and I hope y'all can see that. I will zoom in a little bit on it because so can you see this here? Big Regen Ag Lab in the upper left-hand side. We got PLFA. Okay, I'm getting a thumbs up from Patrick. Very good.
Yes,
Again,
we
I do not necessarily know. I see up here in the upper right-hand side, cluster 64, field 4087. Um, I'm not exactly sure what all of these are, but we're going to go through. There's four samples, obviously, to a cluster. And so I'm going to go through a couple of these. I'm going to walk through what we're looking at and talk a little bit about how we rank these. So I mentioned before that when we extract all the fats, so when I look at this report, everyone says, well, what do you look at first? Right? Like what do you go to first? Well, I go to the overall rank first. So the overall rank is actually a um, it is actually a sum of five of these different things. Okay. And it's weighted. They are a weighted sum. So I'm going to give them to you in order, and then we'll talk about them as individual pieces. So the first thing that goes into the overall rank is the functional group diversity index. That carries the most weight, and it's about 40%. Total biomass is number two, tied with fungal to bacterial ratio, which is down the very first number below the overall rank. Those two items are equal in weight, and I want to say they are 20% each. Um, yeah, 20% each. And then the third thing is the protozoa, or the fourth thing, excuse me, protozoan to bacteria ratio. And that is 10%, something like that. And gram positive to gram negative is the last one. And that's 10%. So we take all of those things together to make this weighted average, if you will, to give us this overall rank. Now, where did the ranking come from? This is where everybody cringes a little bit. I pulled it out of thin air. That's not completely true, but I mean thousands. I mean, I've run over 200,000 PLFAs at this point. And when I was doing my master's research, I ran thousands of them. And I basically built a histogram, bell curve, and figured out I just calculated the average of all these numbers. Now, does that mean the average works for every location and every growing situation? No, not at all. What is truly average for the desert of New Mexico is not the true average of Alberta, Canada. Okay. That information doesn't exist. Guess what, though? Echodiasis has a very large database, and I wouldn't be shocked if that's the kind of stuff that Dr. Lundgren works on, right? Is looking at geographical location, soil type, management, the type of operation you're in. That is how people have developed their own type of rankings and averages to understand this data better. But this gives us a pretty good place to start. And it's something that we've used and we mine our database to develop these averages. Nobody likes to be compared to everyone else, but they always want to know how they're doing. Well, to know how you're doing, we have to say relative to something, right? So let's talk about. So I look at that overall rank first. Okay. Now, obviously, on this one, it says good to very good. We have everything from very poor to below average to um, average, above average, good, very good, and excellent. Okay. It's a pretty wide range because there's so many numbers being put into this to calculate this rank. We didn't want to just say good or bad, right? And I don't like to use the words bad because there are other constraints, environment, et cetera, et cetera. These numbers do change. But let's go through this one. Starting with the functional group diversity index, because I mentioned that that's the most important. That one carries the most weight. So the way we calculate this, if you've ever done any research on diversity, you'll see the term Shannon Weiner. There's a Shannon-Weiner diversity index. And normally that index, so for example, you go out to a forest and you lay down a transect 100 meters long, and you walk down the transect and you count how many different species you see, and then how many individuals of each species you see. So you walk down this transect and you're going to look at animals and you say, okay, I saw rabbits. I saw 100 rabbits. And then I saw raccoon. I saw one raccoon. I saw 13 deer. So the total number of species you saw was three, and then you put in the number or the abundance of each one, and you calculate this diversity index. The index takes into account two things: the number of groups or species represented and their relative abundance to each other. So if you saw 100 rabbits and one raccoon, you score a pretty poor diversity index. If you saw 50 rabbits and 50 raccoons, you score a higher diversity index based on relative abundance to each other. Okay? I did not come up with that equation. I am not a mathematician. That exists, though, and it's the most widely adopted. Now, we're not measuring species, right? We're not measuring species, so we are not giving you a species diversity. We are calculating diversity from the functional groups: bacteria, gram-positive, gram-negative, fungi, mycorrhizal, saprophytic. So you'll see that community breakdown. And on the community breakdown, that is number one, it tells you which groups are represented. And number two, what is their relative abundance? So we put those numbers in and calculate the diversity index. The diversity index operates much like it's a logarithmic scale, which means the range is very small. So from about 1 to 1.6. Yes, we do see samples that score below 1. But the general range is 1 to 1.6. I would say 1.3 is then average. You're getting above average, and you'll see we provide those individual ranks for that. So on this example, we've got good diversity, functional group diversity. Now, a lot of people ask me, well, why does that matter as much as the biomass or things like that? And I said, okay, well, and I'm an analogy person, so here's my analogy. Imagine the community you live in or the community you're closest to. And let's say there's 5 million people that live in Oklahoma City, and every single one of them is a lawyer. So you've got a really high biomass and you got everybody doing the exact same thing. How well does that community function? Not well at all. Everybody just walks by and says hello to each other and hands each other invoices for $400. It it doesn't do any good. So biomass is important, but I would argue that a community of 1,000 people that has teachers, maybe a doctor, police officers, fire, et cetera, that community functions better. Now, ideally, we would like a highly functioning community, but then we would like a community that has enough biomass to function, right? For the work. You've got two firefighters in a town of 50,000 people, you're going to be in trouble if a fire happens, right? So biomass and diversity, but diversity is more important. And if I'm trying to look at things and identify gaps, diversity is what I'm going to work on first. How do we build diversity? Well, through management. So protozoans and fungi, for example, do not like tillage. Tillage cuts up fungi and destroys them, Protozoans don't really care about your tillage equipment. The problem they have is that the soil structure starts to collapse and they swim in soil pore spaces with water. So if we can't get water in the soil and we don't have pore spaces, guess what? Protozoans don't do well. Bacteria don't care. They don't care about your tillage. So now we end up with a bacterial dominated system completely, and our diversity score suffers. Now, I'm not just picking on tillage. Fallow. Fallow is the number one most detrimental thing for mycorrhizal fungi. They rely on a host plant. Without a host plant, they will not survive. So they form spores. And no, we cannot measure spores with PLFA. But then the next year comes around and we plant our crop and the crop starts to grow, and the mycorrhizal fungi have to kind of start over. They have to start their colonization process over. And so we see this lag before they pick back up and grow, etc. So those are the things. If I see a poor diversity score, I'm immediately going to look down at that community breakdown. I'm going to look at which groups am I missing or which groups do I have but are relatively low. Everyone asks me, what should that breakdown be? Like, what should my total bacteria number be? There isn't an answer for that, and it's because it's relative to all the other groups. That's what the diversity score is for. Okay. So if you look at that diversity score and see a 1.487 and say that's pretty good, well, then that's telling you that that is pretty close to where your fungi should be relative to your total biomass and your bacteria, right? Um, the one thing that's missing on this example is protozoans. Now, Lynette even asked me this before we got started is, what's the deal with protozoans? They often come back as zero. The limitation on PLFA testing for protozoans is that we are looking for three specific fatty acids in a world of fatty acids. We identify 160 to 200 different fatty acids, and we're looking for three specific ones to identify protozoans. On top of that, protozoans are predators of bacteria. They graze on bacteria. So their numbers are relatively low compared to bacteria and fungi anyway. That makes it difficult to pick them up. Keep in mind that when you see a zero, it doesn't truly mean that there is not a single protozoan within an acre of your soil. What it means is below detection limit. We were unable to find them. I can't put that on the report because I have to use numbers to calculate things. So zero is what goes in there. It's very common. The other thing that will influence that number is the time of year the samples are taken. These were pulled in about now in mid-May. Um, Everything in the soil has to ramp up. Bacteria ramp up first. When soil conditions start to warm up, when things start to, if we got moisture, resources, bacteria are the opportunists. They go first, followed by fungi, finally followed by protozoans, right? We see the same patterns in nature everywhere. We had a really good year, really good wet spring, lots of food around, et cetera, et cetera. The rabbit population explodes, and then it's 6 months later, a year later, you start to notice there's a bunch of coyotes running around. Well, that's because there's a lag in how long it takes for those to come back or come in. So that's what we're looking at. If my diversity score is good, I don't even look at the community breakdown. I don't need to. It's already telling me that I got a pretty good score. I'm then going to look at the total biomass. This one's above average. Again, I'd love to see it climb up to excellent, but I'm more concerned about the diversity than I am the biomass. That biomass is very likely going to increase. Again, this was taken in May. If you waited another 6 weeks, it would very likely be higher. Um, then we go over to the community ratios, fungi to bacteria. You'll see here that this one is 0.3762, ranks as excellent. We're simply calculating that by taking total bacteria, or sorry, total fungal biomass divided by total bacterial biomass. It's just that ratio between those two groups. The reason why you probably have heard this, you'd like a ratio of 1 to 1, right? That's the idea. PLFA is not going to ever give you a ratio of 1 to 1. We know this, and that's why the ranking on this is weighted. So, for example, if I had 1 pound of bacteria in my hand and I had 1 pound of fungi in my other hand, that is a 1 to 1 ratio based on weight, right? The problem is that when I extract all the fatty acids, the PLFA out of bacteria, they have around 4 times more per pound than fungi. So if I extract all the fatty acids and I use the fatty acids to measure them, it looks like I have 4 times more bacteria than I do fungi, even though I started with 1 pound each. So our cutoff is 0.25. When you get a ratio above 0.25, you're in the good, very good, excellent category. From zero point 0.2 to 0.25 is above average, and it's like 0.15 to zero point 0.2 is average. So when you get above that 0.25, you are sitting closer at that 1 to 1 type of ratio. Now, I had somebody ask me, I've had multiple people ask me this, said, well, then why don't you just, you know, factor that 4 in and show me a ratio here that is actually 1.2 to 1. I did that for a while, but then I had really smart people call me and say, well, I did the math and your math is wrong, right? I took 265 divided by 706 and I don't get 1.2, I get zero point 0.3. I know. So we just took that back out because I created a lot of questions with people that were doing the math on the report. Um, so sorry, so let me go back. The fungi to bacteria ratio is excellent. You jump down to the protozoa to bacteria. Obviously, this one ranks it very poor. It's all bacteria. Now, even though that ranks it very poor, the overall rank on this sample is good to very good, right? I mentioned that the protozoan to bacteria ratio does not carry a ton of weight on that overall score of how well your system's functioning. Protozoans are fickle and they're just hard to measure. So we're not going to base everything on that. Finally, we have the gram positive to gram negative. Quick mention about what that is. Positive does not mean good, negative does not mean bad. And I'm not trying to insult anyone's intelligence. Everybody has a different understanding of bacteria and nomenclature. All bacteria fall into one of those two categories. Gram positive means that they're built like a chicken egg. They have a hard outer shell or a cell wall. Gram negatives have a thinner cell wall, but they have a membrane outside of that, a thin cell wall, and then a membrane inside of that. Why do we care? Gram-positive bacteria are built like castles. When things go bad, it's too dry, too hot, too cold, they go into siege mode. They hide behind the cell wall. They go dormant. They're alive, and we can measure them, but they can wait it out. Gram-negative bacteria don't have that option. If that outer cell membrane gets damaged because of too much heat or it's too dry, they have a tendency to die. So what we use this ratio for is a gauge on your environment, soil environment. And you probably sit there and go, if I could control the rain, we wouldn't have this conversation. I could do whatever I wanted, right? But you control the microclimate with your management. No, you can't control the temperature of the air, but you control the microclimate that these microbes live in. We want this ratio to be between 1 to 1 and 2 to 1. That gram positive always tend to do a little bit better than gram negative in soil environments. You know what we typically see though during prolonged drought? That ratio starts to go up, right? We get to a 2 to 1, 3 to 1, 4 to 1, and in some cases, I've seen it as high as 8 to 1. Bare fallow, summer fallow wheat ground, 105-degree air temperature day after day in Kansas, wind blowing 40 miles an hour, and your ratio gets way out of whack. If you don't think if you don't think you can influence this microenvironment, this is the best example I've ever seen. Jimmy Emmons down in Leedy, Oklahoma, he shows this picture where he took soil temperatures and he was, 5 feet away from each other. And he had a soil temperature on bare soil at like 148 degrees or something like that. And where he had residue and crop, cover crop, the soil temperature was like 86 degrees. And it was over 100 degrees that day out there in Oklahoma. Well, he pulled PLFA samples from those locations and, under the cover crop, this and that. Yeah, there was some drought stress there. Um, but his overall biomass, his diversity, and this ratio between gram positive and gram negative was in the average to above average category across the board. 3 feet away, 4 feet away, he pulls a sample, and that ratio was 7 to 1. Biomass had crashed, diversity had crashed, and there was nothing different, right? Other than just one of them is microclimate managed, the other one is not. So that tells us a little bit about things. I look at that one quite often, and again, early in the year, we've had people pull samples in January before in Nebraska, in North Dakota. Guess what dominates a landscape? Gram-positive bacteria. They can tolerate that cold and things like that better. Doesn't mean they're active. They're just alive. They're there. We can measure them. The stress indicators, I'm only going to mention them. The stress indicators are more useful in controlled research type plots. These are the things that the university people like to look at. Um, they're in a greenhouse, they're in, potted soil studies, et cetera, et cetera. They're adding different things. It's not as big of an influencer at the field scale. Okay. Now you can still glance at them, saturated to unsaturated. We've all heard of saturated fats versus unsaturated fats. Um, Microbes change the composition of those fats based on temperature. So if we see a lot of really high saturated fat numbers, odds are your soil's really hot because they're making saturated fats so they don't melt, right? Saturated fats are more stable at higher temperatures, so they're making more of them so they don't melt. Conversely, if your soil is really, really cold, you're going to see a higher number of unsaturated fats because saturated fats turn into solids when the environment's cold. Mono to poly is just the degree of unsaturation. Are they mono unsaturated or poly unsaturated? It's just a feed off of the ratio above it. The last two are growth indicators. So when microbes are active, bacteria are actively dividing, they make a lot of what's called pre-16 and pre-18. Those are division precursors. When the microbiome is more static or they're not in rapid growth, they produce more cyclo fatty acids because those are more stable. So they convert 16, what they call 16:1 omega-5 cis into 17 cyclo fatty acid. Typically in the spring, again, these were in May, conditions are typically better and the microbes were here and now they're going into active growth phase. So we tend to see this higher number of precursors, and that's why we rank that as active or very active. It tells you that it's in a high growth phase. Um, in the fall, we typically see that the opposite, right? They're in a latent growth phase or non-active growth phase. So you can glance at those, but I do not necessarily directly make management impact decisions on those things. It's the functional group diversity index, the fungi to bacteria ratio, those things that are going to influence more. Um, so I went through how to go through a report. There's multiple reports here. If I go down here to the next page, um, let me go back up here. Good to very good. Notice the biomass 2600, diversity 1.48. We go down here, the biomass has dropped from 29 to 19. The diversity's gone up from 1.4. Again, logarithmic scale, so 1.4647 to 5, 1.5. And the overall rank has gone up from, instead of good to very good, this one's just slightly better at very good. Um, Still have this ideal gram positive to gram negative ratio. Our fungal to bacterial ratio is even higher, up to 0.5. Still don't have any protozoans. Now we've got one here where the diversity is still good. It's still good. Our biomass is excellent. Now, it is more difficult to keep your diversity high when your biomass gets really high. Because again, you can go from a 1500 biomass up to 4000, but if it's all bacteria, your diversity score comes down. Okay. This is a really good example. We started to see protozoans show up in this one, and we have a lot more bacterial biomass or just overall biomass for protozoans to feed on. In other words, we have more rabbits. So now we're getting more coyotes. Protozoans are excellent nutrient cyclers. They consume bacteria, they graze bacteria, and they excrete the excess nutrients that they take in. So a bacteria cell has a carbon to nitrogen ratio of around 3 to 1. Corn, for example, is around 80 to 1 at maturity. What that means is that for every pound of bacteria I have relative to a pound of corn, I have around 25 times more nitrogen tied up in a pound of bacteria than I do a pound of corn. When we fertilize, the microbes take 90% of it. Whether or not they give it back to the plant is relative to how happy they are, how fed they are. And number two, are they being consumed by something else? Protozoans have a carbon to nitrogen ratio of around 15 to 1. What that means is that for every 5 bacteria they consume, they're getting 5 times more nitrogen than they need. Just like the rest of us, like all organisms, when they consume that in excess, they excrete it as a waste product. That process is called mineralization. So protozoans are pooping out nitrogen in the ammonium form. And if the plant can take it up in the ammonium form, great. Some of it goes back into the other microbes. The nice thing is that if this cycle's happening all the time, microbacteria don't live very long, right? A couple weeks. So if this is happening all the time, there's ample opportunity for the plants to siphon off some of this nitrogen, build protein, capture carbon energy, and do what they do to feed back into the system. Okay. Again, still ideal on the gram positive to gram negative. You'll see here, look at that fungal to bacterial ratio. I just said you very rarely see one above one. You're not, I just said you're never going to see one that's 1 to 1. Well, I ate my words on this one because there is one. And honestly, I can't tell you if I've ever seen them. I've seen them get close, usually on compost. So I don't know what in the world they're doing here on this sample. The rest of them look great. This one's exceptional. Um, could just be where the sample was taken. Fungi, they're not, you got to remember, not all of this stuff is just spread evenly across an acre, right? There are hot spots. I always tell people, imagine if you've seen this picture, NASA put it out several years ago. Um, when you look at the Earth from outer space at night, or the United States, you can see New York City, you can see Chicago, you can see Denver, and there's all this black space in between. That is what microbes look like in the soil. If you took a square inch of soil and you could look at it that way and make them light up, that's what it would look like. So they're scattered all over, and it's the pore spaces that connect all of them, much like our interstate system is what connects all of those major areas. So protozoans are like trucks. They drive up and down the interstate. The protozoans are swimming back and forth between these pore spaces, and they're grazing on these colonies of bacteria and excreting nutrients, etc. Sometimes we get into this area where we get a concentrated um, hit. Um, Steve, would you say this test is no indication of soil productivity, but a test to show soil life and makeup? My highest PLFA test came back higher ranked on my worst soil that I'm trying to figure out how to make it more productive. Yes, PLFA does not necessarily coordinate to productivity. Okay. Most of you know that I worked with Dr. Ray Ward or for him for many years. And he used to talk about productivity relative to soil fertility. He said it is difficult to have a soil that is productive that is not also fertile. However, just because a soil is fertile does not mean it's productive. And it's the same to be said with soil health. It is difficult to have a soil that is productive without the proper fertility and without microbes and some degrees of soil health or measurements. But just because we have a soil that is healthy does not necessarily mean it's productive. There are other constraints to that. Interestingly, we see that from time to time, Steve. It's not that uncommon. And part of it is that your productivity constraint on that field may not be related to the soil life in it. It could have other things to do with it, such as pH. There's other, soil structure, which I know you work on all the time. Um, there are a number of factors that go into productivity, um, and not just this. Interestingly, we see soils out of the desert Southwest, and I don't mean desert Southwest of Nebraska. I mean desert Southwest of the United States, and they'll score very well relative to their area, but they're not productive at all. Um, And typically it's physical hindrance of root development, or disease pressure. Because keep in mind, PLFA, we can't differentiate with disease. Um, Fusarium would show up on this test, as a biomass. So not everything we're measuring is butterflies and unicorns. Some of this is witches and devil. We just can't differentiate them with this type of test. The last one here on this example, this kind of falls back into the previous two, or really close to the first one. Again, these are taken down transects. So we had one transect that was the third one here, which kind of stood out with that really high um, score on the fungi to bacteria ratio. But overall, all of these are pretty good. Um, I'm not sure. I was told that one of these groups was from an orchard, I believe. One of these is from a grazing system, and the last one, the last set is from potatoes. Um, Grazing system and orchard are pretty wide open terms. There's a lot of ways to manage those systems, right? Overall, I don't know if this specific example is the grazing system or the orchard. I was able to identify the potato, and I'll jump to that one. But these all look really good, and interestingly, this is the other. So now from cluster 64 to cluster 71, similar, similar numbers. Um, good diversity, very good biomass, excellent. Here's your fungal to bacterial ratio again, pushing one at zero point 0.9. We do have protozoans here, a lot more of them. I know that number looks really small. That ratio number looks really small. That number's never very high, right? It's like counting the number of gazelle on the Serengeti versus the number of lions, and then making a ratio. Um, So same idea. This sample is slightly gram-negative dominated. Uh, that isn't necessarily an issue. Uh, Gram-negative bacteria do a really good job at remediation. Um, the remediation for whether it's heavy metals or pesticides, herbicides, etc. I'm not saying that because gram-negative are there, that's an indicator that those things are also there. I'm just saying you see gram-negative bacteria dominate mining reclamation sites, oil spill sites, etc., etc. Um, Jumping down, biomass dropped off quite a bit here. But you'll notice both of them are very active in the bottom right. I mean, it's up and coming. Still an excellent fungal to bacterial ratio. Diversity is even higher because, again, your total biomass is lower. Your diversity is more likely going to be higher. Again, just because bacteria are the things that tend to come down the most. So again, relative abundance. Very good again. Ideal, excellent, good numbers across the board. Nothing really to point out there. Um, this one is interesting because the diversity, or sorry, the total biomass just plummeted, right? Just plummeted down to 500 in the poor ranking. You'll notice here, though, the overall rank is still good to very good. Again, diversity, fungal to bacterial ratio, etc. Also in a very active growth phase. Don't expect to see protozoans here because the total biomass, the food source, is low. Um, don't have an explanation for that because I don't know where it was taken, but this could be an area that either was relatively waterlogged for a while, or the opposite. Could be incredibly dry, but this could be a low area in the field. That transect was laid down, um, et cetera, et cetera. Those things will hurt biomass. But overall, the management reflection here is that okay, we have some constraints on overall biomass, whatever those may be, but we're not destroying the fungal component. We're not destroying the diversity piece of this. And so the functionality, even though it's reduced, we still have all of the components to make this car drive, right? I don't care what kind of engine you've got in your car. You can have the nicest, biggest supercharged engine on the planet. If you don't have a steering wheel and you don't have four tires, you ain't going nowhere. So in this scenario, we have all the proper components, but it runs a little rough. We need a tune up on the engine. And mostly when that happens, it's environmental. Where we see huge gaps in the community structure, that's usually management. The last one here, this is the one that I identified as potatoes. Um, this piece right here, this sample right here, looks pretty good. Um, Potatoes are tough because of the way they're grown, especially at scale, right? There's just a large amount of disturbance in potatoes. Um, it doesn't Potato growth is also pretty indicative of nematicides, different things that we're putting in. I'm not here to say, you should grow potatoes no-till on a scale and you should do all this and never use these things. I'm just saying there's trade-offs. If we're in that type of system, there are trade-offs that we have. This one does pretty well. This one also looks pretty well or pretty good. And then I think we had a pretty sharp drop off here. So this is an example, poor biomass. There's your diversity down below 1 at zero point 0.9. Um, didn't register any fungi here. Again, that doesn't mean that there isn't a single fungal component out there. It's just they're so low that we weren't able to detect them. You'll notice here, this is very gram-positive dominated soil, very likely highly disturbed. Um, et cetera, et cetera. This is pretty typical of what we see even on like the Michigan sands growing potatoes. Um, Again, I'm not judging, knocking anything. I'm just showing you numbers relative to management. Um, I like potatoes. I eat potatoes. So uh, there are things that you can do, but it's definitely not an orchard, right? It's not managed like an orchard. Think we see this one jumps back up. So overall, this is really good. I don't see potato numbers like this. I really don't. I got 1 out of 4 examples here where this is what I typically see. So pat yourself on the back um, for what you're doing um, in this potato production system because overall this is very good. There's just this one area, and again, this might be an eroded hillside. It might be a washout area. Maybe this is where the trucks are parked or traffic back and forth. There's a lot of factors that can go into that where we might be dealing with compaction. We might be dealing with um, lack of residue here. Maybe this is a staging area, et cetera, et cetera. So one area out of the 4 in the field. Um, but yeah, those were the examples. I do have time. I know we're at time, but I do have time to take some questions. If anybody's got them, I know that was kind of a crash course, but I mean, I'm going to go through every report from every single field exactly the same way, right? So if you're sitting there thinking like, well, I want to go through my report, there you do it the same way. Overall rank, jump over to functional group diversity, biomass, right? If my diversity is really low, I'm going to drop down to the community ratio or the community group. And I'm going to look and see, well, what am I missing? And then ask yourself, well, if I'm missing mycorrhizal fungi, ask the question about the system you're in to think if you understand why. Say, oh, well, I do a lot of fallow, or this sample was taken at the end of the fallow period in May. Maybe that's why. Um, and then it's up to you if you want to do something about it, or if you just say, no, this is why it is. um, that's what I'm dealing with. But I go through every report exactly the same way. Are there any questions? I'm sure plenty of questions. Does anybody want to speak up and ask a question?
Here's your opportunity. Yeah, to be able to ask that question that you've been wondering about. What makes protozoa thrive?
High bacterial biomass and um, good soil structure. Protozoans will move up and down the profile with moisture. Yep. So yeah. So
So less
Matt said so
disturbance.
less disturbance. Yes, that's the first. That would be the first key. The second one. So I used to believe. So I used to think this 5, 10 years ago that tillage was the number one most detrimental thing you could do to your soil system from a soil health perspective. Um, I had it in my mind that a disc blade destroyed soil structure. And then I started measuring aggregates and realized that aggregate stability, when you talk about micro aggregates, we are talking 53 microns to 1.125 micro or millimeters, so 125 microns. Macro aggregates are 125 microns up to 250 microns. I don't care how sharp your disc blades are and how tightly packed together you put them. Those disc blades are not cutting up your aggregates. What tillage does is it oxygenates the soil temporarily, it redistributes carbon, and the bacteria go nuts. It's like giving a 5-year-old a pixie stick. They bounce around the house for an hour and then they crash. But while they're bouncing around the house, they're consuming large amounts of carbon, the carbon that holds together all of the soil particles. So then when it rains, the particles or the wind blows, the particles no longer have the mortar to hold everything together. So imagine going to your church or your school, they're usually built out of brick, and chiseling out all the mortar between all those bricks. Soon as you get a slight disturbance, the whole thing falls apart and collapses on itself. Tillage speeds that process up, but it doesn't directly cause it. The thing that causes it the most is fallow. So microbes will break down and rebuild aggregates on about a 3 week basis. They're remodeling their house every 3 weeks. And we think, man, why every 3 weeks? Well, keep in mind that's like um, 2.5 generations for microbes. So they're remodeling their house every 3 weeks. Well, there was this thing that happened or didn't happen, depending on what side of the fence you're on, but it was called COVID. And when that happened, you couldn't go to the store and buy anything. So imagine you decide you're going to remodel your kitchen. You get out your Sawzall, your sledgehammer, and you break everything down. And then you go to the store to buy new countertops and cabinets, and there's nothing there. How do you rebuild? That's what happens in the fall, especially when we harvest a crop. The microbes continue to break down aggregates, but their store, their shopping center is living plants. That's what's pumping the building blocks back into the soil so they can rebuild what they've destroyed. When that doesn't happen, soil structure starts to fall apart. And I've seen this. I've seen guys that are 30-year no-till, corn, bean, corn, bean, corn, bean with nothing in between. And guess what? The top 3 to 5 inches of their soil is like concrete because the aggregate structure has disappeared, even though they're not doing the tillage. And I know you can sit there and think of countless examples. You've seen the same thing. 7-year alfalfa, 7-year alfalfa stand, walk out in it. Soil tilth is wonderful once you get below the first 4 inches, but usually the top four are pretty rough. What little carbon alfalfa produces, we chop it, bale it, and haul it off, right? So when we say disturbance, it's not just physical disturbance, it's also providing the resources to rebuild that. And that's one of the limitations. Um, Would it be advisable to repeat this test periodically to make comparisons from a baseline? Yes, Rebecca, it is. But as someone else pointed out down below, Bruce, it is important to be mindful of soil temperature and moisture. PLFA is one of those that there are definitely better times to sample. Like if you're in Montana, don't go out in March or sorry, January and get out a drill bit and drill a hole. And I can tell you it's going to look pretty bad. But within that mindset is there's really no best time. The best time is when it works for your operation, but you want to be somewhat consistent. If you're trying to answer the question, how do my soils look in May versus October, then sample those and say, I want to see the difference across that growing season. But if you're trying to go year to year to year, um, if you're going year to year, then you want to be sampling the same time each year. On top of that, I will mention this. This is not a test I recommend that you run every single year on every acre. The test is too expensive. Someone asked what the price is per sample. The price is $90. It is very complex, time consuming. It's an expensive test. The best way this is used is outside of like echodiasis is doing this for research, right? To understand management. They're doing it at a very large scale. For those of you who might want to do this on your own, it's you pick out 2 fields, one of which you've managed very differently than the other. You take one sample from each field and make your comparison that way. You can do it in this field. For example, if you have an area that just struggles and you've got areas that are great, you can do that comparison. Typically, this test is used as a report card, meaning that we're not going to make direct management decisions from this test, meaning in the conventional mindset. Like you're not going to look at this test and go, oh, I've got X amount, or my pH is 5.6. I need to put on X amount of lime. It's not a prescriptive type test. The management decisions you might make from this are going to be large scale, meaning do I till this field or not till this field? Do I want to bridge the fallow gap? I mean, these are larger scale management changes. Um, the Jumping back to Steve asked a question. He said, I have 2 fields that have mycorrhizal fungi, one doesn't. Following 3 different crops, and all were in fallow state. I'm going to add mycofungi to every seed this year. Um, yeah, you said you saw the mycorrhizal was present behind peas and millet, but nothing behind corn. I'm not sure what type of corn you're growing, Steve. And I have been seeing more and more where corn, especially with the genetics that they're putting into corn, and you probably are not growing GMO corn if I had to guess, but I'm just saying, throwing this out there, with a lot of the varieties that they've developed and a lot of the genetics that they've developed and all the different things they put in, we're seeing less and less mycorrhizal colonization and development on some of those varieties because the corn is just not being built for that. It almost gets bred out. You could argue the same thing or show the same thing with soybeans and rhizobium. Um, as they continue down that path. Now, I'm not here to say we shouldn't be growing any of those things, but it is one of the trade-offs that we're starting to see. Kind of like the trade-off when they started making corn stalks as big as tree trunks. They said, well, the corn's getting taller. We got to make a stronger pith and a stronger stalk because it's blowing over because we're putting on 400 pounds of nitrogen and it's growing too tall. And well, yeah, then the trade-off is that guys are saying, well, these corn stalks don't disappear. Well, part of that is you've got to, part of that is biology problem, right? But the other part of it is that your biology was never designed to eat the corn residue from 350 bushel corn in central Iowa when the corn stalks are as big around as your forearm. There's just too much for them to consume at any time, right? So the trade-offs back and forth. I do know millet is a pretty good supporter of mycorrhizal fungi. Um, at least that's what I've heard and seen. Not sure on the peas though. And I will mention this as well. There's a pretty good misconception out there. So you'll hear that brassicas don't support mycorrhizal fungi, right? You mustards, radishes, etc. That is true, but they don't kill them either. So I've had people that are afraid to use brassica because they think that they're going to kill the mycorrhizal fungi. If you plant a monoculture mustard crop, the mycorrhizal fungi essentially see that field as fallow. They just do not form associations with mustard or those types of brassicas. But they aren't killed because of the mustard. They go back because they don't have a host. If you mix in brassicas with oats, sorghum, whatever, the brassicas just ignore the mycorrhizal fungi. They just leave each other alone, and the mycorrhizal fungi forms associations with those other host plants. Okay, so you're not hurting anything by putting brassicas into that group.
And Bruce wants to know, it seems to me that it would be beneficial to know soil temp and moisture of the time of sample.
Yeah,
Is that
I kind of
important to you?
kind of mentioned that with so you want to be mindful. That doesn't mean we need to go out and stick a thermometer in the ground, record the temperature and measure moisture. But you want to be somewhat mindful, meaning I've had people call me and say, "Hey, I pulled a PLFA sample last year at this time. Should I pull it again?" And they'll say the soil temperatures are 20 degrees behind where they were last year. My answer is no. Wait a couple weeks. Let the soils warm up so the temperatures are closer. Or we haven't got any rain or any moisture for the last 4 months. Things are really dry. Don't waste your money. Don't waste your time. Unless, of course, you are trying to answer the question, how has this drought affected my PLFA numbers? If that's your question, pull that sample. If you're just trying to compare to last year because you want to see what your management did, wait until it rains. And I understand some of you go, that might be a long wait. Well, skip that year then. My job, I mean, yes, we get paid to run soil samples, but we do not get paid to take your money so you don't have it to operate your farm. That doesn't do us any good. The data needs to mean something to you.
Yeah. And that is one of the things that we do at ICDICES is we do go ahead and do soil moisture and temperature samples when we're out there
Yes.
sampling on those transects. And that information is going to be available for all of you as we're working on the grower portal. So you're going to actually be able to see that raw data. It's coming. It's just taking a little time to get it all together because we do want to include all the years, especially if you've been sampled multiple years. That way you'll be able to see it all. So yeah.
Yeah. And it's great that you guys are collecting that information because when you're doing this at the scale you're doing it, you don't have the convenience to be at every single location at exactly the right time, right? So by having those temperature differences and moisture differences as part of your data set, you're able to say, hey, these PLFA numbers look maybe quite a bit different, but we understand why because we can look at those temperature and moisture differences and kind of, if you want to kind of say correct for it, you can. But yeah, I know with as far as you're traveling and as with the many samples as you're pulling, it's a little different than going out to the East 40 that's a mile down the road and pulling them when the time's right. Anything else anybody wants to touch on?
Anybody else want to ask a question or if there's anything that you've been curious about? Now's the time.
Doesn't even have to be related
Before
to PLFA if you don't want it to be.
it doesn't, yeah. If you have a question about something else,
And
a
if I
lot of you
don't
have
know, I'll
received.
just say I don't know.
All right, here's one. Yeah, looks like the saturated unsaturated ratio is low, and a question mark. So what could be causing the saturated and unsaturated to be low?
So
And
saturated,
so
unsaturated, low. Um, low, low does not necessarily mean bad. Um, what it means is that those microbes have a high number of unsaturated fats, which means they could partially be cold stressed. Um, so let me go back. The the membrane, this seems weird, but the membrane of the cell has to be what they call fluid. It's not liquid like water, but it has to be somewhat fluid. The only way a microbe can get something in and out of the cell is either you pump it through a channel protein, think of a straw piercing through the membrane, or they bring it across the membrane directly, which means that the membrane has to flow. It has to be fluid. Unsaturated fats, think of vegetable oil, things, olive oil, those are liquid at room temperature, right? Saturated fats like bacon grease, tallow, those are solid at room temperature. So the microbes change the composition of the fats, and it has to do because saturated fats are straight. Each one of these is straight. So they pack very tightly together and they bond with each other and make this rigid type of structure. Unsaturated fats have kinks. They're kinked. I can't really do it that great, but they're kinked up. And so they don't pack as tightly together, which is why they're liquid at room temperature. If your soil's really hot, the microbes produce a ton of these saturated fats because if they don't, they're going to melt. Their membrane literally melts. It becomes too fluid and it breaks and they die. Unsaturated is when it gets warm and they start to make more saturated. It starts to cool down, they make more unsaturated. The other time they'll have a lower ratio is when they're actively dividing. So if you're taking a single cell and splitting it into two cells, you have to take your membrane and pinch it together in the middle and then pop it apart. And so they have their membrane is more fluid in that way. I'm not sure if that answers your question completely, but that they change that composition daily if needed. They're able to change the actual bond structure in those fats. And the way they do it is they kind of pull one out of the membrane, break it down and consume it, take the phosphate group, and then rebuild a new fat on the end of it, and then reinsert it back into the membrane. It's kind of neat how they can mark those and pull them out. Again, because these things change so rapidly at the field level, don't get too bogged down in what those numbers are because those things change pretty quickly and you're not controlling that environment as much. Where you've got people like in a greenhouse, I mean, they're controlling temperature, they're controlling the moisture, they're controlling the CO2 concentration in the air. I mean, they control it all. Um, and they're not even really growing anything in soil. They're growing stuff in a media that they created typically. So, a little different. And they control all those things so that way they can add different plants or they can add different treatments and then see how that influences these types of ratios. But at the field scale, maybe the sun was hitting the soil in that square inch, you know, coming down through your plant leaves and the sun was hitting that spot. Um, Simple as that. Maybe there was a worm casting next to that probe. You know, you stuck the probe in the ground, there was a worm casting in it. I mean, all of those things can influence that environment from a microbe standpoint because bacteria, you know, how many bacteria can you put on the head of a pin? It's such a scale that we have a hard time understanding, let alone managing, right? On that type of scale. You manage the field, but on that scale, it's very difficult. It's nearly impossible.
Awesome. Well, are there any more questions? We have about 2 minutes left. And so if anybody has a question, go ahead, or otherwise we're going to close this one. I want to thank Lance Gunderson for helping us go through this PLFA test. I know I learned a lot because this was one of the tests that, yeah, I just wanted to know more about and where to look and all of those things. And yeah, I think Lance did a great job. And let us know if there's something else that you have questions or whatever. Lance will definitely be able to help you be able to understand it, or just send the questions to me. All of you have my email address. My name is Lynette Miller. And yeah, shoot me the email and I'll tell you what, we'll find out the answer for you. It may take me a couple of days because, yeah, needless to say, Lance is pretty slammed right now, but yeah, I'll get the answers for you. All right.
We'll get
So
them.
thank you all very much. And thank you, Lance.
Yeah, thank you, everybody. Appreciate it.